Building blocks of protein structures. Physics meets biology. The field of molecular biology was born 70 years ago with Linus Pauling's stunning prediction of the two building blocks of protein structures based on preliminary X-ray evidence and quantum chemistry. The first building block is the alpha helix and is a piece of the chain curled up into a spiral conformation. It is topologically one-dimensional. The second building block is an almost planar two-dimensional sheet assembled from zigzag strands. There are two distinct sheet assemblies because of the two different schemes for hydrogen bonds to provide scaffolding. The situation is loosely similar to a neat person packing her clothes by folding them into lower dimensional manifolds and placing them in her suitcase. Here, we revisit this problem and present a physical and mathematical derivation of the same geometries with no adjustable parameters and no chemistry input. We make one constructive hypothesis that the protein maximizes its self-interaction in its native state or ground state and explores the consequences. We do this in the simplest situation of not taking into account the 20 distinct amino acid types. We work instead with a homopolymer, a chain made up of just one type of bead. Here is the summary of the steps involved. We use the simplest correct description of a chain, a uniform tube or garden hose of non-zero thickness. The protein atoms occupy the space within the tube. We maximize the self-interaction of a continuum tube by winding it into a tight helical conformation. The local radius of curvature is set to be equal to the tube radius. A looser turn would leave empty space in the middle of the helix, while tighter turn is forbidden because of static considerations. Successive turns of a helix are placed on top of each other and alongside each other. This allows us to deduce that the pitch to radius ratio of the space-filling continuum tube, a helical tube, helix, takes on the universal value of 2.51 approximately. We then consider a discretized version of the space-filling continuum tube with a bond length of around 3.81 angstroms. We find the rotation angle of the helix, approximately 99.8 degrees, that maximizes the self-interaction. We also find that the tube radius is 2.63 angstroms. From these values, all attributes of the helix can readily be derived. Remarkably, the discreteness offers us with the possibility of existence of a second building block of protein structure. Namely, the discreteness of the tube admits a qualitatively distinct second building block, that is two-dimensional strand when the rotational angle is 180 degrees. We can then work out assembly of strands in two ways to create parallel and anti-parallel beta sheets. These steps lead to a slew of qualitative zero-parameter predictions for the geometries of the building blocks of protein structures. We find the excellent accord between the mathematical predictions and experimental data on high-precision protein structures. This is the summary table of predictions coming from the theory and the experimental data for the alpha helical geometry. In the same way, these are the comparison between theory and experiments for the two possible beta sheet geometries that correspond to parallel and anti-parallel beta sheets. In summary, there are 5 lessons that are learned from our studies. The first one is that discreteness is essential for the diversity of protein structures. Second is that there is a remarkable consilience in the manner in which chemistry and biology provide an almost perfect fit to the dictates of mathematics and physics. Number 3 is that protein structures are not subject to Darwinian evolution. Sequences evolve, as do functionalities, but the native state folds do not. Further, the sequence chooses the best fit fold from the predetermined menu of Platonic folds determined from considerations of geometry. And finally, our study points to the possibility of designing powerful artificial machines and artificial life which do not rely on carbon-based chemistry. Thank you very much.